strand A plus DNA strand B yields DNA strand A, B double helix plus heat, right? Heat is released. It can be considered as one of the products, right? So what happens then if we input heat into this system, into this reaction? What's going to happen? Well, if we consider heat to be one of the products, right? It's not exactly the Ch.8's principle that you would be very disappointed. But instead, right, what we're doing is we're shifting the equilibrium of this reaction backwards. Okay, so by adding heat, we take the DNA double helix AB and form DNA A and DNA B strands, right? This process is called melting or denaturing. By adding heat to break apart two strands, called denaturing. Now we can remove the heat, and again, we saw like in chapter 1, DNA strand A and B will then spontaneously come together, right? So by varying the temperature, we can manipulate if the DNA is single-stranded or double-stranded. And we can see that here, right? How do we measure how much of our, well, actually, let's just go here, right? So as we increase the temperature, we can measure the relative amount, let's say, right? We'll come to absorbance in just a sec. We can measure the relative amount of DNA that is double-stranded or single-stranded, right? So down up here, we have single-stranded DNA. Well, down here, we have double-stranded DNA. So double-stranded to single-stranded DNA. As we add more heat, notice that we start to increase the amount of single-stranded DNA that we have. The temperature at which we have a mixture of 50% single-stranded and 50% double-stranded DNA is called the melting temperature, or the Tm, right? So above the Tm, we can consider the DNA to be mostly melted or mostly denatured. And below the Tm, we can consider the DNA to be mostly annealed, right? And how do we measure how much of DNA is either double-stranded or single-stranded? We'll use ultraviolet spectroscopy, or UV spectroscopy, okay? And this comes down to a very simple principle that DNA and RNA, well, DNA specifically, absorbs light the best at 260 nanometers, which happens to be ultraviolet, right? So if we see over here, we have a sample of DNA, okay, and what we're doing is we're shining a known amount of light through our sample, and by measuring how much light we receive on the other side of that sample, we can take a subtraction and figure out how much light was absorbed by that sample, right? So if we start with a certain amount of DNA, we start shining light at 200 nanometers and move up to 210 and 220 and 230 and so on and so on. We see that we get a peak up here at about 260 nanometers. Means that DNA absorbs light the best at 260 nanometers. But an observation was that this is temperature dependent. In other words, it's dependent on the native state of the DNA. Denatured or single-stranded DNA absorbs light better than double-stranded DNA, right? So here, this blue line corresponds to the absorbance at 260 nanometers of single-stranded DNA or denatured DNA. Whereas here we see that double-stranded DNA is absorbing less light at the same wavelength. Okay, why is this? Well, it's the basis of the DNA that actually absorb the light at 260 nanometers. By having the two strands separated or denatured, now we expose all of the bases to the environment, and there's more base available to absorb this light. Whereas in double-stranded DNA, a lot of the bases are sequestered inside of the DNA double helix. So they kind of cast a shadow. So the ones down here, for example, aren't absorbing as much light as the ones, say, up here or up here, right? They're also protected by the backbone itself, right? So they're able to absorb less light. So what we're looking at here, again, I just told you that down here is the double-stranded DNA and up here is the single-stranded DNA. Really what we're measuring is the relative absorbance at 260 nanometers, okay? So we measure our sample. We see here, right, we'll start at the bottom here, right? So at cold temperature, we say, all right, everything's annealed, that's say 50 degrees, everything's all together in a double helix. We'll just call that 1, okay? And then we find the ratio, how much is this moving up, right? So this is, 105% of what we started with. This is 110% of what we started with, and so on, until we reach some equilibrium where everything is denatured, right? So this is how this was determined. And we can do these experiments now to observe the melting temperature. Now, in reality, we don't ever really observe the melting temperature. We don't do experiments on DNA and say, oh, what's the melting temperature? Instead, what we found is that there is a relationship between the DNA sequence and the melting temperature. Okay, so if we know the sequence of DNA, and we know a bunch of other parameters, the salt concentration and length and things like this, okay, we can estimate the melting temperature so that these kinds of experiments are no longer necessary. So what effect would you think on the melting temperature would occur if you had a DNA double helix that was particularly high in GC base pairs? Would you think it'd be higher? So say that we had two DNA strands of the same length, but strand or double strand A had 20% GC, whereas strand B had 80% GC. Which one is going to have the higher melting temperature?
The GC has higher because it has more hydrogen bonds, so it makes it stronger.
Right, so the more GC base pairs we have, the higher the melting temperature. It means that the higher the temperature we have to put in to separate the two strands. Reason being, again, GC base pairs, recall, have 3 hydrogen bonds between them, while AT base pairs have 2 hydrogen bonds between them. In fact, the genomes of many extremophiles, particularly ones that live in deep sea vents, they have an unusually high proportion of GC base pairs. All right, DNA polymerase extends DNA in the 5' to 3' direction, okay? And it uses nucleoside triphosphate, specifically 5' nucleoside triphosphates, in order to do this. All right, now we're going to cover the actual mechanism in more detail later on in chapter 29. But here again, right, we get binding between the first or the next base, rather, the next base in the template. So on the bottom is our template strand. Up here is the strand that's being synthesized. And what's going to happen? Well, if this is a T, then it's going to base pair with A. So what we have here is 5' ATP. So the substrate is always a nucleoside triphosphate. We'll get base pairing here. Then the 3' hydroxyl of the previous base in the newly synthesized strand performs a nucleophilic attack on the alpha phosphoryl group here. This kicks off these two phosphates as pyrophosphate, and now we have a phosphodiester bond between what used to be the alpha phosphoryl group and the 3' hydroxyl of the previous sugar. This exposes the 3' hydroxyl over here, which is now able to undergo another round of addition, right? So the overall mechanism looks like this. We have our DNA template strand, also called our primer, and we are adding onto the 5' end, or rather the 3' end of our newly synthesized DNA strand. So it's being elongated in the 5' to 3' direction, which is why we refer to DNA in the 5' to 3' direction, the direction in which it's synthesized. Again, the 3' hydroxyl, the exposed 3' hydroxyl, is going to attack the alpha phosphoryl group. This is the alpha, beta, and gamma phosphoryl group. So it attacks the alpha phosphoryl group of the base that is now base pairing with the next exposed base on the template strand. This kicks off these two phosphates, and we lose that as what we call pyrophosphate, right? Now we form our phosphodiester bond, right? So the products of this reaction, right? The nucleophilic attack here, okay, the formation of the phosphodiester bond, and pyrophosphate, this has got a delta G, which is very close to 0. So this process is readily reversible. All right, we can just as easily go from this state to this state as we can from this state to this state, but we don't observe that happening in real life, which is a real problem. Okay, so how do we lock this in the forward direction, right? Because otherwise we'd never be able to synthesize a full strand of DNA or RNA, as we'll see. Well, this comes down to this byproduct here, pyrophosphate. Pyrophosphate, while it is stable in solution in the cell, pyrophosphate never gets to exist for any appreciable amount of time. Because as soon as pyrophosphate is made by any reaction, it's almost immediately hydrolyzed into 2 orthophosphates, right? And this releases 19 kilojoules per mole, standard free energy, right? So what we have here is an example of a coupled reaction, right? And a coupled reaction are just 2 reactions that happen one after another, essentially. And so we can add them together. That is great. Anyway, so we have, say, here, a DNTP plus DNTP plus DNA of N length right? What we get out of it is a pyrophosphate plus DNA of N plus 1 length, right? Then this pyrophosphate immediately is hydrolyzed into 2 orthophosphates. So we can add these together. We can cancel out the pyrophosphates because they are on either side of the yield side, right? So it's a product and then a substrate, so they cancel out. So the overall reaction here is DNTP plus DNA of N plus H2O yields DNA of N plus 1 plus 2 orthophosphate. This is our balanced reaction, and when we have a coupled reaction in this way, we can do something to their delta G's, right? We can add them together. So if the delta G here for this reaction is about 0, and the delta G for this reaction is negative 19 kilojoules per mole, well, now we can add them together so that the overall delta G naught prime of the balanced reaction is negative 19 kilojoules per mole, which of course is favorable and irreversible, right, because it's far from 0. So the hydrolysis of pyrophosphate is what drives this reaction forward, and that's a theme that we're going to see in multiple different types of reactions. The hydrolysis of pyrophosphate drives specific reactions forward, okay, by contributing that negative 19 kilojoules per mole. We'll see this in RNA and DNA synthesis. We'll see this in RNA synthesis, DNA synthesis, glycogen synthesis. Yeah, glycogen synthesis. Many different reactions are driven forward by pyrophosphate hydrolysis. I'm not going to spend time talking about RNA genomes because we have 15 minutes left. Now, RNA polymerase works much the same as DNA polymerase. In fact, the mechanism is almost identical. Right here we have a DNA template strand, but what we're synthesizing here is RNA, not DNA. Okay? But the mechanism, notice, is exactly the same. The 3' hydroxyl, which is also present on 2' deoxyribose, right, attacks the alpha phosphoryl group, kicks off the pyrophosphate. Now we get our phosphodiester bond again. Because this reaction is essentially identical to the one that we just talked about for DNA, the delta G here is close to 0, but we still get the hydrolysis of pyrophosphate, blocking this in the forward direction. The difference, the main difference between DNA polymerase and RNA polymerase then, is that RNA polymerase does not require what we call a primer, while DNA polymerase does. Okay, and what does that mean? A primer is something that DNA polymerase has to add onto, right? So it can only add onto an already existing nucleotide polymer. So it needs a 3' hydroxyl, like we have over here, right? It needs this 3' hydroxyl in order to add anything. So it can't start synthesizing a DNA strand de novo, right? So if we imagine that we have just a DNA strand, nothing else, right? So here's our template DNA strand, 3' to 5', and we want to synthesize a new strand, 5' to 3'. Well, the first base that's going to be added here, okay, DNA polymerase can't add because there is no 3' hydroxyl before it to perform that attack, right? So DNA polymerase needs a primer, right? So if we have a situation like this, and what we want to synthesize is this stretch of DNA going forward, that's possible because over here we have a 3' OH, right? This over here is what we call the primer. Right? DNA polymerase needs a primer. RNA polymerase, though, does not require a primer. So it can do that first situation. It can add the first nucleotide and let's just go. Right? So RNA polymerase does not require a primer. Though they do both require, obviously, a template, specifically a DNA template. Now, when we're talking about RNA synthesis, we can break down the 2 strands of DNA that it's using, right, into different names, right? Obviously, it's only going to use one of the strands as a template, right? So here we have our mRNA that is synthesized, okay? And it's using the template strand as the template, right? And we usually call this either the template strand or the minus strand. The other strand, though, is the coding strand, or the plus strand. Even though this strand down here in black is not participating at all in this reaction. So why do we call this the coding strand? Well, we can see that if this is being used as a template, right, then the sequence of messenger RNA that's generated, or RNA in general that's generated, must be complementary to the template strand. This template strand is also complementary to the other strand in the double-stranded DNA. So this and this are complementary, and this are complementary. That means that this and this must have exactly the same sequence, with the exception, obviously, that the thymines are replaced with uracils in RNA. So the strand that does not participate at all in the synthesis of the messenger RNA molecule, we call the coding strand, because if we look at the sequence of that strand, that is the sequence of the messenger RNA that's going to be synthesized. But the template strand, or minus strand, is the strand that's actually participating in the synthesis of that messenger RNA molecule. All right. Now, where does transcription begin? Transcription begins in eukaryotes at the Pribnow box, and this at the promoter site, right? And so what we see is something that looks like this. We have the start of RNA transcription. Question is, right, how does RNA polymerase know that it should start here and not just somewhere else? Well, it comes down to these two regions here, right? We denote transcription starting at the +1. This is the first base that's going to be present in the messenger RNA. So this is the +1 position, followed by the +2 position, +3, 4, 5, and so on. The base directly before the first base is the -1 position. Now, of course, this is not going to be part of the messenger RNA, right? But we still do care about it in the DNA, because it's at these upstream regions or negative regions that the RNA polymerase is going to recognize, oh, this is a gene. And we have the -10 element and the -35 element, meaning that over here, right in the middle of here, thereabouts, is the -10 position, so it's 10 base pairs away from the +1, and the same over here, the -35 element. These two elements together are recognized by RNA polymerase, and it sits RNA polymerase just far enough away from the +1 position so that by the time that RNA polymerase dissociates from the enzyme that actually recognizes this, it's sitting right at the +1 position and can begin transcribing that gene at the correct site, right? Now, eukaryotic promoter sites are a little bit more complicated. Okay, here we have analogous regions. Instead of -10, we have the -25. Here, instead of -35, we have -75. Right? These serve similar roles, okay, but the regulation of transcription in eukaryotes is significantly more complicated. We have insulators, enhancers, things like that. We're not going to get into that because it's not really relevant. But in both cases, right, we do need some region of the DNA that RNA polymerase recognizes that is just upstream of the transcriptional start site. Now, transcriptional termination, we'll talk about later on in the semester, so I'm just going to skip this, right? Because, in fact, let's see what we got here. All right, so from slide 25, you're going to write this down in your notes. From slide 25 to slide 28, we're going to skip, because we're going to talk about this in way more detail in chapters 28 through 30, so it really makes no sense to go over it twice. So from slides 25 to 28, we're going to skip over those. I do want to gently touch on splicing, however. Once messenger RNA is synthesized, right, it can undergo transformations, specifically splicing, okay? Now, splicing just describes this process by which a region of messenger RNA is excised from that messenger RNA, right? And how was this discovered? Well, it was discovered that if you take the DNA double strand that codes for a specific gene, let's say gene X, and you take the messenger RNA for gene X, and you anneal them together, okay, you get a strand that is displaced, right? Or displaced down here, because now you're forming a DNA RNA hybrid. Okay, so this is our template strand, this is our coding strand, and we get an interaction between the messenger RNA and the template strand of DNA. What was found in eukaryotes when this same experiment was replicated? This is from bacteria. In eukaryotes, okay, what was found is that we get the formation of these loops when that occurs. So we get interactions between the messenger RNA and the template strand. Then we don't get any interactions for a while, and then we get interactions again, right? So we get this displacement loop here, right? This doesn't bind to the messenger RNA. This then, this sequence on the DNA, which must have been part of the messenger RNA at some point, was excised from the messenger RNA, right? So we call that an intron, the INT standing for intervening. It's an intervening sequence in the messenger RNA, right? And we can see why. And here we're getting interaction here, and to synthesize the messenger RNA, right, we must have synthesized starting from, let's say, over here, all the way over here, right? We must have synthesized at some point a messenger RNA that included all of this sequence. But the mature messenger RNA, now we see, doesn't include this sequence here because it doesn't base pair with it. So what happened to it? Well, the messenger RNA must have had it excised. It couldn't have been excised from the DNA because it's still present. We can still see it in the loop. This piece that was excised from the messenger RNA is our intron, right? So for example, right, this is the beta globin gene. The beta globin gene is synthesized like this, but it's got two introns, meaning that if we take the beta globin messenger RNA, right, the beta globin messenger RNA, and we hybridize it with the DNA double helix containing that gene, it will have two of these kinds of loops. Those two are our introns, right? So here and here we have an intron in the beta globin gene. These two are cut out of the pre-messenger RNA, right? So we get transcription, we form the loops here, okay, in the messenger RNA. Those are cut out by a machinery in the nucleus called the spliceosome, and this segment here is annealed, or ligated rather, to this segment. This segment is ligated to this segment to make the full beta globin gene, right? Many, in fact most, eukaryotic genes are spliced, right? And you might ask yourself, what's the point of this? Why not just synthesize the gene and be done with it? This allows for the complexity of our proteome, right? In other words, the number of proteins that we can make compared to our genome, right? Does anyone know how many genes a human has, roundabout?
27,000?
Yeah, about 20,000, right? Does anyone know how many different types of proteins we can make? A lot more. Yeah, it's about 100,000. So how can we make 100,000 proteins from only 20,000 genes? Well, this is a phenomenon called differential splicing or alternative splicing, right? So here's a very basic example. Say that there's an antibody, right? And there's two types of antibodies. There's ones that are membrane bound, right, on the surface of B cells, and there's ones that are free to exist in the serum, right, to bind to antigen just in the blood, right? So we have to have one gene that codes for an antibody with a little bit of extra on the end that anchors it to the membrane, as we see here. And we have to have a second gene that codes for the same antibody, just without the little bit that anchors it to the membrane. And now we've essentially duplicated that antibody. That's a waste of space, both information-wise and space-wise, and energy-wise, because we're essentially encoding the same information twice, right, wasteful. So instead, what happens? Well, we have the gene that codes for this antibody, right? But there's an intron in the middle, and this intron, right, separates the antibody part from the membrane anchoring part. If we don't splice that out, right, well now we have this over here, right? So we have this over here if we don't splice, and we have it over here if we do splice. So now we're able to make two different proteins from one gene, right? This is alternative splicing. This is how the complexity of our proteome, the number and identity of proteins that we can make, is so much larger than just the number of genes that we have, because we can splice them, cut them up in different ways in order to perform different functions, right, depending on whatever that particular cell needs, right? Good, so we'll end it there, and I will see you all on Tuesday. Have a good weekend.